Hi, I'm Wannes, and I would like to present to you our study published in NPJ Biofilms and Microbiomes on how known and novel probiotics can affect periodontal biofilms with a focus on strain-specific effects. As you may know, pathologies such as periodontitis are characterized by a dysbiosis or a disbalance between the host and the microbiome, which can maybe be solved by the use of antimicrobials. However though, since the use of antimicrobials can be quite contentious, alternatives such as probiotics are being investigated to see if you can reintroduce beneficial species to then change the balance between the host and its microbiome back to a healthy state. Now, while the use of probiotics is becoming quite well known, there are a few important issues that need to be thoroughly investigated. One such issue is the variations in effectiveness between probiotic species, but also between strains of the species. Looking at the case of Lactobacillus plantarum, we know that there are some strains that can be quite effective for irritable bowel syndrome, while others can be ineffective or some can even make it worse. This then poses the question, how about probiotics for oral health? Are these effects also there? Together with a bunch of wonderful people, we investigated 3 known probiotics as well as 6 novel probiotics. To do this, we used several methods, starting with an agar spotting technique in which the pathobion's growth can be inhibited by the probiotic. On the other hand, we also used some more advanced techniques such as the flow cytometry and a biofilm model. For the flow cytometric approach, we created a cell-free supernatant of the probiotic, in which pathobionts were then incubated. The effect of the probiotic supernatant was then evaluated through flow cytometry using a viability treatment that contains a general dye as well as a viability dye that only enters dead cells, which allows us to clearly characterize living versus dead cells. Using our biofilm model, in which we establish a community of several well-known oral bacteria, we then form biofilms with the probiotic on hydroxyapatite, which after growing can be recovered and treated also with the viability dye to specifically look at the DNA of living cells through viability qPCR. While the agar technique has the benefit of being quite low tech, the other techniques are a bit more quantitative but slightly harder to perform. For the proximal inhibition on agar, we saw that few of the strains had significant inhibition on the growth of the pathobionts, while others had a more moderate effect, and some of the presumed to be effective probiotics didn't even show any inhibition. To confirm this, we then also evaluated them in the flow cytometric approach, by first characterizing all of the pathobionts, which allows for a quick and very quantitative determination of the pathobiont's viability when exposed to the supernatant of the probiotics. So, when comparing the viability of the pathobionts when in spent medium against the probiotics, we saw that several of the probiotics affected the viability of the pathobionts, indicating that they contain antimicrobials that affect membrane integrity. On the other hand, some of the strains had much more moderate effects or didn't even affect some of the pathobionts. To see if the flow cytometric approach reflects what happens in biofilms, we then also tested that with the biofilm model. And what we saw is that some of the probiotics were able to reduce the total biofilm, while others were not. And using species-specific primers, we also evaluated how much probiotic was in the biofilm. And while we hypothesized that more probiotic in the biofilm could have more effects on the total biofilm, this was not what we observed. To understand why this might be the case, we looked at the individual species, in which we saw again the known species being quite effective, but also some of the novel species, which was in line with the flow cytometric approach, although the magnitude was slightly different. Another benefit of the biofilm model is that it also allows us to look at the effects on commensal species, which many of the Lactobacillus probiotics strongly affected. On the other hand though, the Streptococcus salivarius probiotics showed a much more moderate effect. So, when keeping the total biofilm in the back of our head, we can also see that there are some proportional changes caused by each of the probiotics. Some of the probiotics were quite prevalent in the biofilm, which is quite interesting, but also had some unexpected effects. But the Streptococcus salivarius probiotics seemed to only affect the periodontal pathobionts, but not each of them was as effective. So, to conclude, the flow cytometric approach served as a rapid and quantitative screening method, mimicking the effects in the biofilms, some differences in magnitude. Perhaps the most important takeaway is that there were very strain and species-specific effects on both the pathobionts and the total biofilm. While generalizing this is maybe somewhat contradictory, we observed that several of the Lactobacillus probiotics had effects on both pathobionts and commensals, while the Strepsilivarius species were more specific but did not affect Aggregatibacter actinomycetum comedans. Finally, probiotic prevalence did not correlate with their antimicrobial effects. That's all. Thanks for listening.